Good afternoon. Welcome back. Today we're going to continue our discussion of antiviral responses, and we're going to talk about adaptive immunity. Just to remind you, we've talked so far about two kinds of host defenses: the intrinsic defenses that are always present in the uninfected cell, and they comprise apoptosis, autophagy, RNA silencing, antiviral proteins, and the innate immune system, which is induced by infection. Today we're going to talk about the adaptive immune system, which I mentioned before is tailored to the specific pathogen and which also is characterized by memory. To remind you of how the adaptive response is initiated, on the left, we have an epithelial cell sheet. It's being infected by viruses. These viruses have entered. They started the reproduction cycle. The cells have sensed infection via the innate immune system, and the cells respond by the production of cytokines, these red dots which are secreted from the cell, and they attract cells like dendritic cells and macrophages. Here we're showing an immature dendritic cell being attracted to the cytokines. It undergoes maturation. It will then travel to the lymph node where it'll encounter T cells and initiate the adaptive response. So today we're going to talk a little bit about what goes on in the lymph node and what comes out of the lymph node in the form of antibodies and cytotoxic T lymphocytes. These are the two main effectors of the adaptive response that we're going to talk about, and how they interfere with virus reproduction. First, let's define the cells that we're going to be talking about today. A leukocyte is a general term for a white blood cell, and that includes lymphocytes, neutrophils, eosinophils, and macrophages. So when we say leukocyte, that's what we're referring to. Lymphocyte is a subset of leukocyte, and that includes T and B and NK cells. And these are different because these cells have variable antigen-detecting cell surface receptors. All right, so that's leukocytes and lymphocytes. All cells within the blood are derived from a common precursor, the multipotential hematopoietic stem cell. This is a self-renewing stem cell population which exists in the bone marrow, and it gives rise to two additional precursors. One is called the common myeloid progenitor, and that differentiates further into red blood cells or erythrocytes, mast cells, and myeloblast. This should be myeloblast, and that in turn gives rise to basophils, neutrophils, eosinophils, and monocytes. The other branch is called the common lymphoid progenitor. This differentiates into natural killer cells, T lymphocytes, and B lymphocytes. And some of these are the players that we'll be talking about today, of course. Last time we talked about how the innate immune response instructs adaptive immunity. And let's review that and take it a little bit further today. Remember, we have a virus infection going on in our example that we showed just a little while ago. It's infection of an epithelial sheet, could be anywhere in the body. The cytokines produced have attracted immature dendritic cells to the infected area, and these cells bear a number of receptors, such as toll-like receptors, cytokine receptors. They have MHC class II in their endosomes. They can secrete interferon. They can take up dead and dying cells. They can be stimulated by inflammatory cytokines, and they can detect viruses and viral proteins, nucleic acids, via toll-like receptors and cytosolic helicases. If these dendritic cells detect foreign PAMPs, pathogen-associated molecular patterns, if they detect those PAMPs, they will become activated or matured. They migrate to the lymph node, where they're going to then interact with naive T cells. And so this activation or maturation is stimulated by the presence of virus and viral PAMPs stimulated by cytokines. In the process, NF-kappa B is activated, and these cells look very different when they're activated. Now the MHC class 2 is on the surface where it's presenting viral peptides, and we'll talk about this process in a moment. These are peptides that have been taken up from the exterior of the cell, viral proteins. Of course, they may not be viral proteins, they may be cell proteins. The DCs are always picking up proteins and bringing them to the lymph node. If they happen to be self-peptides or self-proteins, then nothing further happens. But if they're foreign, then we initiate an adaptive response. In the lymph node, the dendritic cell will present these peptides in the context of class 2 molecules. They will be recognized or interrogated by naive T cells. If they are foreign, the T cells become activated and progress further, as you'll see in a bit. This activation requires not only recognition of the peptide in MHC2 by the T cell receptor, but also a co-stimulatory molecule, CD28, and cytokines produced by the dendritic cell. This MHC class 2 molecule is present primarily on mature dendritic cells on the surface, macrophages, and B lymphocytes primarily. And in contrast, class 1 is on nearly all cells, and you'll see what happens with class 1 later on. So let's talk about the way the antigen is taken up by the dendritic cell and presented on the surface. So this is called the exogenous antigen presentation pathway. It involves MHC class 2 again, which is typically restricted to antigen-presenting cells like dendritic cells. So these cells are good at taking up material. They are good at endocytosis and phagocytosis. So here is a protein released from a virus-infected cell, say. It's taken up by endocytosis, proceeds through early and late endosomes. It is then digested in the late endosome into peptides. Meanwhile, the MHC class 2 molecule is translated in the rough ER. It's transported through the Golgi apparatus and eventually reaches the late endosome, where it picks up a peptide, and then the MHC2 molecule with the peptide is transported to the surface. So now we have MHC2 on the cell surface with a peptide, the orange block. And so if this is a dendritic cell, that's the process that's happening in the dendritic cell to present the antigen. It's part of the maturation of the dendritic cell. And then the dendritic cell goes into the lymph node where it finds naive T cells. T cells will interrogate the peptide in the context of MHC2. And if it's foreign, the T cell will recognize it as such and become activated, as we'll see in a moment. So that's the pathway for exogenous antigen presentation. There's a close-up of MHC class 2 molecule. It's a transmembrane protein with a groove in it that holds the peptide so it can be interrogated by the T cell receptor. As you will see, as we've seen before, viruses interfere with all sorts of intrinsic and innate defenses. They also interfere with adaptive defenses. And here's our first example. Human cytomegalovirus interferes with the transcription of MHC class 2 molecules so they don't get displayed on the surface, and the cell will not be recognized as infected by virtue of not having a viral peptide for a T cell to react with. We'll see more examples of antigen presentation being interfered with in a bit. The interrogation of the antigen presentation occurs in the lymph node, as I told you. Let's take a look at that in some detail. We have an extensive lymph system consisting of lymph nodes and lymph capillaries and lymph vessels, which are scattered throughout the body, as you can see here. And the purpose of that extensive lymph system, of course, is to provide entry points for antigen-presenting cells so that wherever there's an infection, the dendritic cells can enter the lymph system, find a local lymph node, and there are local lymph nodes everywhere. And have T cells interrogate their peptides that are being presented. And here's an individual lymph node to show you how it's constructed. There are afferent vessels that bring in the lymph from the circulation. There are efferent vessels which bring it out again. The lymph from extracellular spaces and tissues carries antigens and antigen-presenting cells like dendritic cells and macrophages to the lymph nodes through these afferent vessels. And not just dendritic cells, but antigen is going to go in here because that's going to interact with B cells, as we'll see in a moment. We also have a blood supply for the lymph node to help support the tissues that are present. And these also, of course, permit mixing of blood cells with those that traveled from the site of infection through the lymph. And here in the B cell areas and the T cell areas of the lymph node is where much of the early steps in stimulation of the adaptive response are going to take place. Typically, in us, any antigen is only recognized by between 1 in 10,000 and 1 in 100,000 B or T cells. There are many B and T cells on us, but very few are actually specific for any particular given antigen. However, when a foreign antigen is encountered by a dendritic cell and is presented to a T cell in the lymph node, within 1 to 2 weeks, you have 1,000 to 50,000-fold amplification of those specific T cells and B cells. So if you're infected with a virus and a dendritic cell is picking up viral antigens, presenting them to T cells in the lymph node, the T cells are getting activated. They proliferate extensively. And that's why your lymph nodes swell when you have a virus infection. It's a process called lymphadenopathy. And if you have an upper respiratory tract infection, the lymph nodes around the tract are going to swell, particularly the ones in your neck, just under your throat area there. You can actually feel them. There's one on either side of your trachea, and you can palpate them and see that they're swollen. They'll be tender. They'll clearly be you can clearly able to see that they're swollen, and you can tell if you have an infection or not. The others that are more internal are harder to feel, obviously, because they're buried in deep tissues and so forth. There's just a few on your surface that, and you're not going to have many infections away from the upper tract. If you have a skin infection, you may see local lymph nodes swelling and so forth. But that's the most prominent one where you can see clear lymphadenopathy. There are antigen-presenting cells patrolling all throughout your body, as we talked about later. And here are two examples of very specific immune systems in the gut and the skin, the mucosal and the cutaneous immune system. So here on the left is the gut-associated immune system. Here's the lumen of the intestine. Here's the intestinal epithelium on the inside here, lining the lumen of the gut. And below it are the subepithelial tissues, of course, in an area we call the lamina propria. And this is where there are, of course, blood vessels and lymphatic vessels, and antigen-presenting cells are present. If there's an infection of this epithelial sheet, those cells will produce cytokines. They'll attract the APCs. The APCs will pick up antigens and then get into the lymphatic vessels or blood vessels, make their way to a lymph node. But also, the M cells, which are interspersed among the cells, the mucosal epithelial cells, they transfer antigens across into the lamina propria, and the areas called Peyer's patches, collections of lymphoid cells, and the antigen-presenting cells will sample that too, just to make sure there's nothing going on in the lumen, as well as the actual epithelial cells themselves. So that's the intestinal immune system, or part of it, and you'll see two different terms that are used to describe it: GALT, or gut-associated lymphoid tissue, or MALT, mucosa-associated lymphoid tissue. And they typically refer to these collections of lymphoid cells called Peyer's patches. On the right is a diagram of the cutaneous immune system, or skin-associated lymphoid tissue, or SALT, S-A-L-T. This is made up of keratinocytes and Langerhans cells. Keratinocytes are skin-resident phagocytes. They take up things. They can also secrete various cytokines, including TNF-alpha, IL-1, and IL-6. They also synthesize both class 1 and class 2 on the surface, and they can present antigens to T and B cells if they're stimulated by interferon gamma. Langerhans cells are migratory dendritic cells. They are the major antigen-presenting cells in the epidermis when products of viral infections in the skin are detected. So the skin would be up on the top here, or right here where the epidermis we're in. The dead layer is above it. Here's the actual epidermis itself. When products of viral infections are detected there, these Langerhans cells secrete type 1 interferon. They mature. They migrate to the local draining lymph node, and there they present viral peptides on both class 1 and 2 proteins to antigen-specific T cells. So here, the Langerhans is going to get into a blood vessel and migrate to a lymph node. Of course, they could also get into a lymph capillary and get there as well. So 2 examples of specific parts of the immune system. Our first question is, what is a property of innate instruction of adaptive immunity? A, presentation of viral peptides on MHC2 to CD4 cells. B, endocytosis of viral proteins. C, activation of dendritic cells by cytokines. D, sensing by toll-like receptors. E, all of the above. The answer is E, all of the above. They're all correct. They're all things that we have just discussed in the last couple of minutes. All right, we have the dendritic cell or antigen-presenting cell going into the lymph node. Let's talk about what happens next. Just to back up a bit here, we have T cell precursors on the top right here. They are produced in the bone marrow. They come out of the bone marrow into the circulation as naive T cells. They then go to the thymus, where they further mature into T helper precursors and cytotoxic T lymphocyte precursors. And of course, in the lymph node, as we saw, the antigen-presenting cells, dendritic cells, will present antigens to naive T cells. They will then become activated or mature and then participate in immune reactions. And we have 2 different kinds of T cells that we're going to talk about here, the T helper cells and the cytotoxic T lymphocytes. And the cytotoxic T lymphocytes, or CTL, will be involved in the killing of virus-infected cells. And so some T cell CTL precursor in the lymph node, if it sees a viral peptide presented by a dendritic cell, it's going to become activated. It will leave, go to the infected area, and lyse virus-infected cells. We'll talk about that in some detail in a moment. The other kind of T helper cell, which will also mature after contact with foreign peptides, are the T helper cells. And here you can see this is T helper. It's a CD4 positive cell, has a CD4 molecule on the surface. The CTLs, in contrast, have CD8 molecules on their surface. These are canonical proteins that in part define these 2 different cell types. The CD4 or T helper cells produce cytokines. And depending on what kind of cytokines they produce, they can produce Th1 cytokines, which help the development of CTLs, or they can produce Th2 cytokines, which help in the development of antibodies. So on the right is the cellular arm, the helper cells and the effector T cells. We will talk about how CTLs work in a moment. But let's talk about how antibodies are made. Antibodies are made by B cells. B cells recognize a foreign antigen. They differentiate into plasma cells. The plasma cells secrete antibodies, which then help to counter the infection. We will look at how antibodies work in just a moment. But let's take a closer look at how B cells are activated. when antigen binds to a B cell, B cells display antibodies on their surface that are specific for antigens. When antigen binds, it causes clustering of these antibodies, also called B cell receptors. That initiates a signal transduction cascade, which leads to the activation of this B cell and the production of soluble antibodies. Also needed for that process, the peptides. These peptides are internalized. They're processed. They're bound to MHC class II and put on the surface. Those are then recognized by T cell receptors, by a T helper cell, and specifically a Th2 CD4 positive T cell. That recognition of the peptide by the T cell receptor leads to a signaling cascade, the transcription of genes that lead to the production of cytokines. They're secreted from the CD4 Th2 cell, and they also participate in the activation of the B cell, so that the end result is this B cell, which you can go back here and look at. It becomes a plasma cell, and the plasma cell then secretes tons and tons of antibodies. A lot of antibodies, not tons, of course. So that's the process. These are the effectors. We're going to look at cellular effectors. Effectors meaning what does the job at the end, the cytotoxic T lymphocyte and the antibodies produced by B cells. Let's first look at antibodies. Antibodies, of course, are Y-shaped proteins made of 4 polypeptide chains. We have heavy and we have light chains. Heavy are the longer ones, the light chains the shorter ones. And the top part of the antibody, called the Fab fragment, that's the part that binds the antigen. The lower part, the Fc portion, binds a receptor and has other functions. The antibody binds to antigen via the top part of the molecule, in particular by these hypervariable region, which for each antibody molecule recognize a very specific epitope, a short sequence on a protein, or a conformational epitope, if you will. We'll talk more about epitopes when we talk about vaccines. So that's the antibody that is produced by the B cell after it's activated by the binding of an antigen to the B cell receptor. On the right is a typical time course of production of antibodies in the serum. So on the y-axis, we're looking at serum antibody titer. On the x-axis, we're looking at weeks after immunization. So here we're immunizing the animal with antigen A. Within 2 to 4 weeks, we have an antibody response, which we can measure in the serum using specific assays. That antibody response peaks, and then at about 5 or 6 weeks, it goes down. There's a level above baseline of the antibody was quite low, but there are antibodies present for long periods. If you then wait some time and inject this animal with antigen A again, you will have a quicker and more substantial secondary response. So not only does the response happen in shorter time, but the antibody titer is higher than the primary antibody response, as you can see. And that's because there are memory B cells that remained after the first response that are quickly activated. Making that first response requires recombination and rearrangement of antibody genes, a subject covered in any really good immunology course, and often somatic mutation of the antibody genes to give a high affinity antibody, and that takes time. But the cells that are produced by that, they remain as memory B cells, and so when the antigen is encountered again, you can have a very rapid and more substantial response. Even that memory response will decline in time. You don't need to keep producing a lot of antibodies when the infection is cleared. Now, in this experiment, the second immunization with antigen A, we also mixed in some antigen B, and you can see you have a typical primary response to antigen B. So if the antigen's not been seen before, you will have a primary response which takes longer and which is of lower titer than the memory response. There are a number of different kinds of antibodies that we produce. and they're shown here in this lovely diagram. Start here with IgA, which is a typical antibody found in mucosal layers, like mucus is full of it, secretory antibody. And this is a dimer of two antibody molecules that are joined by a protein called the joining chain, and which also have a secretory protein wrapped around them. And these are secreted into mucus on the mucosal surfaces to protect you from viruses that infect that way, respiratory tract, urogenital tract, gastrointestinal tract. They are protected by secretory IgA. There's also IgD, which is the antibody on the surface of the B cell. There's IgE, which is involved in allergy responses. And then, of course, IgG, the major systemic antibody that we produce against viruses and other pathogens. And then IgM, also a major antibody, but this is the primary antibody response. When you're infected with a virus, the IgM is the first antibody that is made before the B cells have had time to undergo somatic mutation, for example, and selection for higher affinity IgG antibody. Because there are 5 IgM molecules joined in this complex, these individual antibodies may have low affinity for the antigen, but together they have a high avidity, so it makes up for not having the optimal antibody to bind the antigen. So that's produced first, and then it's followed by IgG. After it's gone through all the somatic mutation, recombination, class switching, somatic mutation, you now have a high affinity IgG molecule. And that change is shown at the bottom here. Here's an experiment where an animal is immunized with poliovirus, and we're measuring antibody to poliovirus with days after immunization. And you can see the first antibody to arise is IgM. Very quickly rises to peak titer and then declines by about 20 or 30 days after immunization. And then it's followed by IgG, the green line. Comes up a little slower, but then rises to higher titers and sustains longer. So the IgM is typically gone by 60 days or so after immunization, and it's replaced by IgG. So if you measure both IgG and IgM titers to, say, a virus, you can get a sense for where you are in the infection. If you have if you have just IgM, you're very early. If you have IgG and IgM, you're somewhere in the middle. And if there's no IgM, only IgG, the infection happened a while ago, probably 2 months or more ago. And here we also have the kinetics of production of IgA, which are even more delayed and reach lower titers. How does IgA get to mucosal surfaces is shown on this slide. So here's an epithelial sheet. This could be the gut lumen, or it could be the respiratory tract lumen on the top. Here's an epithelial cell, one, and then it's joined to the next on either side by tight junctions. That's the apical surface, the basolateral surface. And of course, underneath is the lamina propria. IgA is made by plasma cells, just like other antibodies. It's secreted from the plasma cell as a dimer, as we saw previously. It's called polymeric IgA. It binds the polymeric Ig receptor, immunoglobulin receptor, on the basolateral side of the epithelial cell. It's taken up by endocytosis. The endosomes move through the entire cell in a process called transcytosis, and then the vesicles fuse at the apical surface of the cell and release the secretory IgA. This whole transcytosis occurs as the antibody is bound to the PIG receptor, and then the receptor is left behind at the surface. It's cleaved by a protease, and that releases a secretory antibody. And then, of course, the antibody remains in the mucus where it can bind viruses that are coming in. In the context of the current pandemic by SARS-CoV-2, this discussion is relevant because now, here it's March, we're a few months into the outbreak, and many companies are developing tests to detect antibodies against the virus to find out who's been infected or not. Remember, initially the infection is being detected by polymerase chain reaction assays. And those are good as long as the virus is in you, but as soon as the virus is cleared, a PCR will be negative. But how do you know if someone's been infected or not? Well, you can look for antibodies, and you can look for both IgM and IgG, and in fact, that's what these assays do. And these are essentially, this is one released by Alpha Biolabs in the UK, and it's designed so that you could do it yourself. It just comes with a little needle. You do a pinprick of your finger, and a drop of blood goes onto this lateral flow device. You put your blood at one end here. You put a little buffer. The buffer wicks through with the blood, and then you have a result after just 15 minutes. And the result is very cool. There's a control line, which shows you the assay is working. And then there's lines for both IgG and IgM. So here is a negative result. You haven't been infected with SARS-CoV-2. Here you're only IgM positive, which means you're really early in the infection. Here you're IgG positive only, which means the infection has been at least 2 months ago. And both you're somewhere in between 0 and 2 months, of course. It's a very cool assay. It's going to be very expensive. I think this is 300 euros in the UK, just crazy. Hopefully the price will come down, or people are designing these here in the US, of course. They'll be cheaper as well. Now, the way these work, we talked about actually in lecture number 2, the lateral flow assay, where you have a wicking pad of an absorbent material, and you place your sample at one end, and it wicks up, and in the process of moving, the molecules engage reporters. So this figure is from lecture 2. It was actually for detecting antigens in the clinical sample. But here we're looking for antibodies. So we can easily modify this figure, though. We can have, instead of a droplet with antigens, this would be a clinical sample with antibody. That would be a drop of your blood that you're collecting, would be placed right on there. So when you put the drop in that part of the well, that's where it's going. And then in our original assay on this first area, we had antibodies to the antigen. Well, now, since we're looking for antibodies, we're going to have antigen on the pad here. So if you have any, and they could be linked to colloidal gold, so you can see the line or whatever makes the line. So you put viral antigen here, which you have to make and purify. So now if you have antibodies in the sample, the antibodies will bind the antigen. Then the antibody-antigen complex will move up, and you will have, instead of one line of antibodies, you'll have two. You'll have one line of antibodies that will bind IgM, and then you'll have a second line of antibodies that will bind only IgG. They will bind the Fc portion of the antibody that's present in your blood. Those antibodies will have been captured by antigen present here. So as the antigen is moving down with your antibodies bound, if they're IgM, they will be bound there. If they are, the IgM is not shown here. We're modifying this assay. It'll be bound here. If they're IgG, they'll be bound in the second line. And this is your control line, which shows the assay is working. So this will be positive no matter what. So that's how you read this out. You would modify our original lateral flow assay to produce this. So that's an example of what's going on now in the middle of this pandemic, to try and see who's infected and not. Because if you have antibodies, it means you're protected. You don't have to worry, and you can go work. And if we know more healthcare workers, for example, who were antibody positive, that would be great, because we wouldn't worry so much about their protection. All right, back to antibodies. What do they do, actually? Well, an important part of the antibody response is the production of what we call neutralizing antibodies. These are antibodies that block infection. That's what the word neutralizing means. These are an essential defense against many virus infections. They bind to virus particles in the blood or in tissues, mucosal epithelium, wherever, and they prevent spread of the virus. They will prevent infection, and so you block the infection. And mucosal surfaces, of course, it would be Secretory IgA, have the same effect. So in general, we think that most antibodies prevent infection. So if you're vaccinated, you're going to make antibodies. You're going to have a memory antibody presence. And then when you get infected with the real virus, you have a big antibody response that protects you from infection. But we also know that sometimes antibodies, neutralizing antibodies, can be important for recovery. We can give infected people antibodies, and they will recover as well. Here's a neutralization assay to make it clear what I'm talking about. And this is how you would measure the potency of a neutralizing antibody. The lateral flow assay that I showed you, that assay does not necessarily measure neutralizing antibodies. It just measures antibodies that bind to the virus, or the viral protein, actually. This assay measures antibodies that block infection. So these are plaque assay plates. And you can see lovely plaques. I don't think they're real. I think this is a fake assay. We have 11 plaques that we've added. On the bottom, we've had, say, a serum from an uninfected animal. We've made dilutions 1 to 10, 100, 1,000, 10,000. Same number of plaques on every plate. On the top, we take some serum from an animal that has been immunized with the virus. And here's the lowest dilution, 1 to 10. There are no plaques. The antibody is blocking infection. The way this assay works is you mix dilutions of the antibody with a virus preparation here to give you 11 plaques. You incubate it so the antibodies will bind the virus, and then you do a plaque assay. So 1 to 10, no plaques. 1 to 100, no plaques. And finally, when you dilute it out, now you have a couple of plaques, and you see 1 to 10,000, there's no more antibody left. So you can actually use this to measure an antibody titer. And beyond the rapid dipstick antibody assays, which tell you if you're infected or not, have antibodies or not, you don't know the titer from that. This kind of assay can give you the titer, which is important in knowing, how well you've responded to infection, for example. If you want to do a vaccine trial, you should measure neutralizing antibody titers. Here's an experiment that shows that if you give antibodies to an experimental animal, you can protect against infection. And this is an experiment where animals have been infected with poliovirus, and we're looking at percent paralyzed here. And the control animals which didn't receive any antibodies, 100% are paralyzed. And then we have high dilution of antibody here, and then less and less diluted. So this is a little confusing the way this is written, but this is the lowest dilution, the most concentrated antibody, and then diluted more and more from right to left. So when you have a high dilution, most of the animals are paralyzed. Then less and less dilution. They're protected until at the highest dilution of serum, they're all protected. And this is an experiment where you infect the animals and let them incubate for a while, then give them antibody. So you can see you can protect against infection. And this, of course, is the basis for vaccination, but it's also the basis for passive immune or passive antibody therapy. And the key here is knowing how long after infection could you give antibody to someone passively and still protect them. And that differs according to the virus. And so you would have to do a trial to figure out what that interval is. You can't go forever. If you go too far into an infection, it's already almost over. So giving antibody won't help. So you have to catch people early. The reason this is relevant is because this is currently being considered as a therapy for COVID-19 caused by SARS-CoV-2. And here's the logic. You have a patient, a COVID-19 patient who recovers. That patient now has antibodies against the virus, which has helped in his or her recovery. You take some blood, you purify the antibodies from the patient, and then you can give that to someone else. You can either give it prophylactically, and that would be, say, to a healthcare worker to protect them from infection, or to a patient who's already infected to help reverse the infection. And how much you have to give has to be tested. How long it lasts, well, probably not more than a month or two. You could test that as well. And the key in the patient who's already infected, How long after the initial infection can you give the antibody and still have an effect? And those are all questions that are being addressed now. These volunteers are being sought, and these antibodies are being purified, and the trials are going to be starting, if not started already. Now, this is being done now because it's an emergency, and it can be done, and it works. We've done this for other virus infections. I'll tell you an example later in the vaccine lecture. But it's not going to be used forever, because at some point we will actually be able to take blood and purify B cells and clone the genes for individual antibodies, human antibodies from those B cells, purify them, and give them to people as highly purified and highly effective preparations. So that's something that will take longer, because they have to be tried in a phase 1 and a phase 2 and a phase 3 trial. But those have been developed, for example, for Ebola viruses, and they're very effective at reversing infection when given by a certain time. All right, how do neutralizing antibodies work? So there are a number of ways that they work to prevent infection of a cell, and they're shown here. So here's a rhinovirus bound by neutralizing antibodies. And those antibodies could work in a number of ways. They could block attachment. You have enough antibodies bound at the receptor binding site, the virus can't bind to its cell surface receptor, so they can block attachment. But that's not the only way they work. The antibodies can also allow attachment but block endocytosis. They might allow, when bound, attachment and endocytosis, but block uncoating, the release of the nucleic acid. Different mechanisms. Sometimes the antibodies cause aggregation of the virus particle, which prevents them from infecting. And in some cases, there is neutralization after replication. There are a number of different ways where that can happen, neutralization after replication. Let me show you one example here. So here we have a cell that's been infected. So the virus has gotten in, and the virus is going through its reproductive cycle. And, this is an envelope virus, so viral glycoproteins shown in red here are being put into the plasma membrane. Eventually, these will be sites of budding for new virus particles to be made. All right, if in this individual you have an antibody response, these are antibodies against that viral glycoprotein, they will bind to the glycoprotein on the cell surface. And here's one mechanism by which that antibody can block infection. It will kill the cell. This is called antibody-dependent cellular cytotoxicity. And this involves NK cells, natural killer cells, which are part of the innate immune response. We encountered this last time. NK cells have Fc receptors on their cell surface. Those are the yellow molecules. CD16 is a typical one. That is a receptor for the Fc portion of the antibody. So the NK cell recognizes that an antibody is bound to this cell by virtue of the antibody binding the viral glycoprotein. When the Fc receptor engages the Fc of the antibody, that causes release of packets of protein from the NK cell, a process called degranulation. All these little brown vesicles are holding proteins, which we'll talk about in a moment. When we talk about cytotoxic T cells, they are released and they bind to the target infected cell and cause it to die by apoptosis. All right, so that's just one mechanism by which an antibody can cause neutralization of infectivity after infection has already occurred of a single cell. There are other ways as well, but this really gives you a good example. Now, how do antibodies bind to virus particles? Here's an example. This is poliovirus bound to a single monoclonal antibody. I shouldn't say single. There are 60 of them here. It's a monoclonal antibody that recognizes a single epitope, a short amino acid sequence, 8 amino acids or so, on the surface of the virus particle. Polioviruses are icosahedrally ordered, as you might remember. And in this picture, here's a 5-fold axis of symmetry with 5 copies of VP1 around it. And this in purple is the monoclonal antibody. You can see there are 5 copies of the monoclonal binding around the 5-fold axis of symmetry. And because there are 12 five-fold axes, there's 60 antibodies bound. And this is a picture I have on my cell phone, actually. It's beautiful. It's also on the cover of Principles of Virology. So antibodies bind virus particles, and they can block attachment, they can block endocytosis, they can block uncoating, etc. by doing this. As you might guess, viruses evade antibodies. Yes, they do. There are many ways, and we'll come back to this in future lectures. Just want to give you two examples here of how viruses evade antibodies. First of all, there's one strategy exemplified by rhinovirus. There are over 100 what we call serotypes of rhinoviruses. What is a serotype? Let's say we have 100 rhinovirus of these serotypes. We take serotype 1 and you get infected by it. You make a nice antibody response, and next year you get infected with serotype 1 again. You're protected from disease. But if you get infected with serotype 2 or 3 or 4 or all the way up to 100, you get sick because the antibodies against one do not block infection with the other serotypes. That's what a serotype is. It makes antibodies that doesn't block infection with other serotypes. So the strategy for rhinovirus evasion is that it exists in over 100 serotypes. So in your lifetime, you're likely to get many, many rhinovirus infections because you're never going to see all 100. And that's a great strategy. Makes vaccine development challenging. We still do not have a rhinovirus vaccine. But maybe we will someday. It's going to have to protect against all the serotypes. Now, in contrast, poliovirus exists only as 3 serotypes. And for most of the years that we have been using polio vaccines since 1954, that vaccine has worked against all 3 serotypes. They haven't changed substantially. There's one example of the virus changing to escape antibodies, and that's in this blog post. If you're interested, you can check it out. But it involves single amino acid changes in the sites where the antibodies bind, which reduce their binding ability. So that's one strategy for evasion. The other is exemplified by influenza virus, which can change from year to year to evade antibodies. And here's the hemagglutinin glycoprotein of the virus, which is embedded in the membrane of the virus. And this is the top, which attaches to a sialic acid cell receptor. And this molecule is very important for immunity. Antibodies that block infection are directed against this molecule. And in fact, the sites where those antibodies bind, the HA, are shown in different colors here with these interesting names. The ones at the top, particularly orange, green, and blue, these are sites, again, they all are sites that bind single antibodies or monoclonal antibodies. One amino acid change in those sites will reduce the binding of the antibody or block it entirely. And that's why from year to year, if you have a one amino acid change in these sites in the hemagglutinin of influenza virus, the vaccine won't work, and we have to change the vaccine in that particular year. So we have to do surveillance and check very carefully for changes. So that's challenging because you have to make new vaccines almost on a yearly basis because of these highly variable sites. So this strategy here is not to make 100 serotypes, but simply to make one that changes from year to year slightly. Now, as you'll see when we talk about vaccines, the purple and the red sites don't change as much. They are conserved, and so we're trying to make vaccines called universal flu vaccines against those epitopes that won't have to be changed every year. In contrast, coronaviruses don't seem to change by either mechanism. There's certainly not 100 serotypes, and it doesn't seem to be the influenza type drift, antigenic drift, we call that. So one vaccine should be sufficient, as it has been for poliovirus and measles virus and many other viruses. Our next question, which statement about antiviral antibodies is wrong? A, they are important for protection against viral infections. B, they only neutralize virus infectivity. C, they may block virus attachment to cells. D, they can be found at mucosal surfaces. E, IgM is the first to appear, and then IgG, which is wrong. Okay, here's the answer. If you don't want to hear it yet, pause. They only neutralize virus infectivity. B, it's wrong. The wrong answer is B. They only neutralize virus infectivity. There are non-neutralizing antibodies that just bind the virus particle, and those we can detect by rapid assays, but they don't block infectivity. They have other functions. Some of them can bind particles. They can bind, say, glycoproteins on the cell surface. They don't have to be neutralizing and can allow for NK cell killing. Neutralizing has a very specific meaning. It means blocking infection. All right, that's antibody, one half of the adaptive arm. And here's the second half, cell-mediated immunity. And here we think this is essential for clearing most viral infections. Clearing, not preventing, although there's certainly some examples where a cellular response can also prevent infection. And here, what happens is the CTL, the cytotoxic T lymphocyte, which is a CD8 positive lymphocyte, forms a synapse with the target cell. They get very tight up against each other. The target cell is lysed and it's dead, so the virus can't replicate anymore. So we are actually killing our own cells, and that causes pathology, as you might guess, some immune pathology, which we'll talk about next time. But in the process, maybe you limit virus infection and clear the infection. Of course, there are countermeasures. There's always a countermeasure. Always. This process, this recognition of an infected cell by a CTL, requires the pathway of endogenous antigen presentation, which is mediated by MHC class 1 molecules. In contrast to MHC2, MHC1 are present on most cells. And that's good because most cells can be infected. And what happens here is that you have on the bottom your infected cell. A virus has bound it, entered it, starts to make viral proteins. Some of these viral proteins are put through the proteasome. They're ubiquitinated, and they go through this garbage grinder, really, which chops it up into peptides, those orange blocks, which are eventually going to be in MHC1 on the cell surface. But to get there, they first have to be imported into the endoplasmic reticulum by this transporter called TAP1 and TAP2. It stands for transporter for antigen presentation. So the peptide made in the proteasome gets imported into the ER. Meanwhile, the genes for MHC class 1 are being transcribed in the nucleus. The mRNAs come out. They're translated into MHC protein, put up in the ER, and this has a variety of subunits associated with it. Here's the mature class 1 protein. Peptide is loaded. Then the peptide MHC1 gets transported up and put in the plasma membrane. And there, it can be recognized by a cytotoxic T lymphocyte that's been made in the lymph node via interaction with a antigen-presenting cell presenting the virus-specific peptide. That selects for the production of CTLs specific for peptides. They then go out into circulation, find infected cells. They find them by binding their T-cell receptor to the peptide in the MHC1 molecule. And then this infected cell will be killed, essentially. And so again, there is MHC class 1 in the plasma membrane with the peptide in a groove in the molecule. That's what the T-cell receptor sees. So that's the endogenous antigen presentation pathway. Of course, viruses antagonize this. And there are a couple of ways they can do that. They're shown here by these red circles. These are just a few examples. These are typically encoded in genomes of herpes viruses. Human cytomegalovirus, herpes simplex virus, Epstein-Barr virus. These are viruses that we'll encounter in a few lectures. They cause persistent infections. They're not efficiently cleared by immune responses, and in part it's because they antagonize CTL killing of infected cells. So these circles simply tell you different steps where virus proteins antagonize this endogenous antigen presentation pathway. They can interfere with proteolysis of the proteins, the production of peptides. They can interfere with the tap function, the importing of the peptide into the ER. They can interfere with the loading of the peptide in MHC. They can interfere with the production of MHC in the ER. They can even interfere with its transport to the cell surface, which isn't shown here. So lots of ways to prevent MHC from getting on the surface and saying this cell is infected. And if there's no MHC, no peptide, it could be infected and the CTL would not even know it, so it would never kill it. So those are brilliant ways to avoid being killed. And they happen. Here's some counters, some other counters besides those three that I showed you. Here's the pathway that's interfered with, and here's the viral protein that does it. Just to remind you, here's the endogenous presentation pathway that we just went through. And so the synthesis of MHC is interfered with by a VPU protein of lentiviruses. The transporter for antigen presentation, its synthesis can be interfered with by a variety of viral proteins, Epstein-Barr virus, cytomegalovirus. Most of these are these herpes viruses, a few others, HIV and adenovirus thrown in. The function, the importer function of the tap is interfered with by viral proteins. And then we have a variety of ways to fool with MHC1 transport. You can have the protein retained in the ER and never get out by these proteins. You can have it dislocated to the cytoplasm. Some of these viral proteins cause the MHC to be pulled out of the ER into the cytoplasm, and there, of course, it's useless. Can't ever get to the plasma membrane. And then sometimes the viral protein lets all of this happen. You get MHC1 on the surface with a peptide, and then it's pulled into the cell and degraded. Increase MHC1 endocytosis. So a couple of the devious ways that viruses counter MHC1 so infected cells are not killed. How are cells killed, by the way? How are infected cells killed by CTLs? We looked at the mechanism of specificity. How does it work? Well, there are two mechanisms. Again, here's the overview. We have a CTL. It's a CD8 positive T cell, or CTL. It's identifying a virus-infected cell by virtue of a virus peptide being displayed on the surface in the context of MHC1. That interaction can be helped by interferons that are produced from virus-infected cells. That interaction leads to the production of effectors, perforin and granzyme B, that will kill the cell. Perforin and granzyme B, these are some of the things that are released by the NK cells that we showed earlier doing antibody-dependent cytotoxicity. And so first, the granzyme B is a protein shown here in red. Goes into the cell so it's secreted by the CTL, the little red guys here. It goes into the virus-infected cell via a perforin pore that is set up. These are perforin molecules, these purple hot dogs. They're going to make a pore, and then through that pore, granzyme B goes. Granzyme B activates apoptosis. It activates pro-caspases to active caspases, and the cell dies by programmed cell death. So that's the mechanism of killing. These purple hot dogs can also make lytic pores in the membrane where the intracellular contents go out, and that helps to kill the cell as well. So two mechanisms: release of cytoplasmic contents through these lytic pores and apoptosis stimulated by granzyme B. That's how CTLs kill infected cells. It's a very effective mechanism, and that's why many viruses antagonize it. Okay, we've talked about antibodies and CTLs as contributing to prevention of infection recovery. So how do you know what an antibody does versus a cell? So here's an experiment that asks which is important in protecting monkeys from monkeypox infection. So antibody versus cellular immunity. So what we do here is we vaccinate animals with the virus, with an activated form of the virus. And then we do some kind of immune manipulation. We measure neutralizing antibody 22 days later, and then we challenge them with virus, infectious virus, and then see who survives fatality. All right, so The top line is no immune manipulation. We get, again, these are all vaccinated, all three groups. Get nice neutralizing antibody titers, which you can measure by neutralization, right, in a plaque assay. So this is the inverse of the dilution that gives you maximum neutralization, or the last bit of neutralization. After these dilutions, you don't get any more neutralization. And then you infect them on day 28. So you can see 3 weeks it takes to get a nice antibody response, and there's no fatality. So the antibody response protects you, presumably, against infection. But of course, we don't know, because we know there are T cells being made, CTLs being matured here. So how can we tell the difference? Well, we can't. We can't say it's antibody. So let's do an experiment to do that. So in the second group of animals, we deplete them of B cells after vaccination. And you can see they have very low neutralizing antibody titers. We challenge them, and 3 out of 4 die. The numbers are low here because these are non-human primates. They're very expensive. And then, so this tells us that probably antibodies are important for protection. So let's take out the CD8 cells just for the heck of it. Deplete the CD8 cells from the animals after immunization. You get pretty good antibody titers, so CD8 depletion is not really affecting that. And that's not surprising, because CD4 T cells are the ones needed for antibody production. And then you infect them, and they're all protected. So this would tell us that CD8 CTLs are not needed for protection against monkeypox infection, but you could find a virus where they are important. So we have some animals, some infections, antibodies are important. Some infections, CTLs are important. How is it determined which response to make against an infection? Well, that begins in the lymph tissues, where sentinel cells like dendritic cells go in and they interact with T cells. Antigen comes in and interacts with B cells. And they say, here are the peptide. You figure out whether we should make antibodies or CTLs. Antibodies or CTLs. So it starts there. But what's the actual information? It's the decision is made by helper cells, T helper cells. These the sentinels and the macrophages are bearing antigens. They go into the lymph nodes. They make contact with T cells, and these pairs of cells exchange information in the form of peptides and cytokines. Remember, we're starting with a naive T helper cell. Here we go. It's naive CD4 positive T cell. CD4 is a helper cell, as opposed to the CD8s, which are CTLs. So these are encountering antigen being presented to them on sentinel cell surfaces. And then depending on the antigen and the cytokines that are produced, different kinds of helper T cells are made from the naive CD4 T cells. So this differentiation, we're showing 4 kinds of T helper cells here, Th1, Th2, 17, and Tregs. And for today, our purposes, we're going to mainly look at Th1 and 2. But these cytokines that are produced by the sentinel cells are what are driving these differentiations into different kinds. So, for example, Th1 cells are induced in the presence of IL-12, and that induces this transcription factor called T-bet, and that makes a Th1 cell, which is going to give you help in the production of CTLs, CD8 positive cytotoxic T lymphocytes. Production of IL-4 is leading to the synthesis of a transcription factor GATA3, which causes the maturation of these into Th2 cells, and that's the kind that makes antibodies. It stimulates B cells to make antibodies. And these other T helpers are important in tumor immunity and suppression of immune responses, T-reg cells. So on the right is an example of how this works. Here we have a dendritic cell, which is interacting with a virus or a viral peptide via a toll-like receptor. And that particular interaction gives rise to the production of IL-12. So IL-12 drives the production of T helper cells, which will eventually give you Th1 cells, which will eventually give you the production of CTLs. So the IL-12 response is dependent on the interaction of the dendritic cell with the viral protein. So here we have specifically producing CTLs. And if IL-4 had been made from a different virus interacting with the sentinel, you would make Th2s, which would give you antibody responses. So that's what determines the production of antibodies or T cells. In most cases, both are produced, but one may predominate over the other at different times in infection. Our last question for today is, for some infections, CTLs are more important for protection than antibody. How is the CTL antibody balance determined? A, by B lymphocytes, B, by intrinsic defenses, C, by autophagy of infected cells, D, by the mix of peptides and cytokines presented by DCs, or it depends on whether the capsid is icosahedral or helical. The answer is D, by the mix of peptides and cytokines presented by DCs. That determines whether you get the right TH subset differentiation to make antibodies or CTLs. Last thing I want to talk about is memory. These adaptive responses provide memory. And that means if you're infected by the same virus, the response will be rapid and specific, as we showed earlier with that graph. And this is in contrast to innate responses, which typically don't have memory. They have to be reinduced each time, and they're not specific for the pathogen. Memory, of course, is the basis for vaccination. When we immunize you with a vaccine, we count on memory giving you lifelong protection against the pathogen. The idea that infection provides memory was first established back in the 1800s on these remote islands to the north of the United Kingdom, the Faroe Islands. And there was an outbreak of measles there in 1781. This was studied in the late 1800s by an epidemiologist, because he wasn't around in 1781, but he studied this retrospectively. For the next by looking at records on the island, for the next 65 years, the islands had no measles. Now remember, this is a long time ago, and there's not much traffic. There's no airfare. There's no air flights to this island because there are no airplanes. And then every now and then there's a boat. But apparently for 65 years, no measles. But in 1846, there was another outbreak. And so probably by then there were enough people who had been born that were no longer immune. And when the ships brought in people infected with measles virus, they started an outbreak. But interestingly, none of the people who survived the 1781 epidemic, some of those were still around, none of those were infected. It's a long time. And the epidemiologist said, wow, there's immune memory. It lasts a long time and probably doesn't need re-exposure to virus, because he thought people weren't bringing much virus in over the years. But we have now better evidence that you don't need re-exposure to virus to maintain memory. So that's the first clue that infection provides memory. And now, of course, we use this in our vaccines. Here's a graphical display of immunological memory. We have, again, an infection of an animal, and we're measuring on the y-axis either antibody or T cells. Can be either. You get an infection, you have an initial immune response, which takes 7 to 14 days to maximize, and then the response declines. And again, it can be antibodies or cells, T cells. And then eventually you go back to baseline or near baseline. Very low levels of antibodies and T cells here, very few T cells, but you have memory in the form of cells, as we'll see. And if you're then infected years later, right, so now between 2 and 3 years later, you have another infection, you very rapidly make high levels of the immune effectors, either antibodies or T cells. And that either prevents infection or gives you a very mild or inapparent infection. So that's memory. The fact that your immune system can remember this initial encounter and respond very quickly and substantially many years later. And for many viruses, the memory is a lifetime, but not for all. For some, the memory is very poor. And we're just beginning to understand why that is. So the components of memory are both T and B cells, of course. And remember, B cells were activated by antigen along with helper cells to become plasma cells that secrete a ton of antibody. Huge numbers, again, not a ton, huge numbers of antibodies, many, many thousands of molecules per minute. And then those antibodies can affect virus infection. So in the memory B cells exist in the spleen and lymph nodes. They don't produce antibodies. So they're B cells here that have already encountered antigen. So they're ready to go. They have the receptor for the antigen on the cell surface, and you get it infected, and they can rapidly proliferate. So that's one form of memory B cell, but they're also long-lived plasma cells in the bone marrow. These guys down here, they're not making antibodies. They're quiescent in the bone marrow. And again, when the antigen comes around again, those can be very rapidly stimulated. They don't have to go through the whole maturation process. All right, so that's B cells. We also have memory T cells. Here's a graph showing how that works. Here we're looking at the population size of various T cells with days after infection. And this is basically the induction and contraction of the T cell response to a typical acute infection. So you have the infection beginning. You have amplification from a naive T cell, which encounters the antigen in a lymph node. It's amplified to form a number of T cell types. And so you have effector T cells, those are the orange ones, which are going to do something. They're going to be either, they could be CTLs, for example. So they expand and they go and they kill virus-infected cells. But at the same time, you're expanding the different kinds of memory cells, the pink and the green and the blue. And then as the infection proceeds, the infection is controlled, the number of effector T cells declines and eventually goes to zero or close to zero. You can see that illustrated here. The effector cells are going away, but the memory cells are maintained. So the memory cell population, those different colors, are maintained. They're established early on after infection, but they're maintained at higher levels in the blood. And those are the memory T cells. They're different kinds. As you can see here, there are circulating memory T cells, effector memory T cells, and central memory T cells. And then we have non-circulating resident memory T cells. These resident ones stay in specific tissues. They all have lung resident memory cells and so forth that are specific for the tissue. And these are ones that are circulating. They have different markers that allow them to be put into different classes. For example, the effector memory are found in various tissues and the peripheral circulation. The central memory are found in lymph nodes and peripheral circulation. So these are what persist at a very low level in the blood. They weren't there before the infection. So now you have a T cell memory, which can be amplified very quickly when you encounter another of the same virus that's specific for these T cells. Finally, I want to remind you that inflammation integrates. It provides integration and synergy of all these arms of the immune system. Here we have, again, our cell surface or body surface. Could be the epithelium. Here we have the subepithelial tissues, a blood vessel here. Viral infection triggers a local inflammatory response. These cells sense infection. They produce cytokines and chemokines, which attract, of course, dendritic cells and other antigen-presenting cells so that they will pick up antigen and bring it to the lymph nodes. The chemokines also attract other immune cells like neutrophils. Here are some T cells coming out of the blood vessel to come in even and see what's going on. So these signals, the chemokines attracting cells, the induction of adhesion molecules on the blood vessels, which you talked about last time, these cells can then get into the infected area from the blood, and this area, of course, becomes inflamed, as you can see here, swelling, redness, pain, and heat by the influx of fluid in all of these cells in here. And the inflammation, remember, is good. It means there's a good communication between the innate and adaptive responses. The dendritic cells will be recruited. They will bring antigens to the lymph node and initiate an adaptive response, as we've talked about today. And these other cell types can work on the infected cells as well. All right, so that completes our very brief tour of viral defenses. It's by necessity brief because this is an entire virology course. Of course, there are entire courses about immunology and entire courses of specific parts of immunology. But for us, this is enough to get us to the point where now we can return to viral pathogenesis and consider how disease occurs, so we can look not only at viral contributions, but contributions of the immune response. And so next time we'll look at mechanisms of pathogenesis.